Hello, this is Graham Hatfull, and I'm pleased to give you a quick video tour of this paper, Prophage-Mediated Defense Against Viral Attack and Viral Counter-Defense, published in Nature Microbiology. So the context of this paper is in thinking about the dynamic relationship between bacteria and their bacteriophages. Bacteria are constantly being attacked by viruses in the environment, and therefore there is a strong selection for resistance or for mechanisms that defend against these types of attacks, and of course the need for the bacteriophages to co-evolve so as to be able to maintain the ability to infect and to reproduce. It's known that bacterial systems encode viral defense systems. Restriction modification and CRISPR-Cas are just two such examples, and one of the key points of this paper is that temperate phages with the ability to form lysogens, where a prophage is being carried around by the bacterial host, that those prophages can encode defense systems, and because prophages are very prevalent in sequenced bacterial genomes, there's likely to be a large number of new and different types of defense systems encoded by temperophages that are waiting to be explored, and we describe just a few systems in a set of mycobacteriophages in this paper. So the C-Phages program is an integrated research education program offering authentic research experiences to undergraduate, primarily first-year students in phage discovery, phage isolation, characterization, and genomic analysis. And this program has generated a large collection of sequenced phages, notably about 1,200 phages that are genomically characterized and are known to infect a single common host, Mycobacterium smegmatis strain MC² 155. These phages are genomically diverse. They can be grouped into different groups or clusters according to their nucleotide sequence similarity, so phages within a cluster are related to each other but unrelated to phages in different clusters. And the stars of this paper are the phages that constitute cluster N. There's a total of 11 of them, 10 of which we've characterized in further detail. They all have similar cipher-viral morphologies as shown by these electron micrographs. They have extensive DNA sequence similarity, and you can see their relationship here according to a network representation of their gene content. If you align the genomes of the cluster N phages, you can see that they're very similar to each other. They're especially closely related in these virion structure and assembly genes in the left parts of the genomes, but in the central and right parts they tend to have these characteristic mosaic relationships where there are segments of close similarity between genomes interspersed with segments of much greater divergence. If we look at the genome of Michel Mabel a little more closely, you can see here again in the left arm, these are the virion structure and assembly genes, head and tail genes. These are followed by the lysis cassette, nearby the immunity cassette containing the integrase and the immunity repressor, and then in the right arm are these early lytic genes of whom the functions, many of these genes we don't yet know. The phages are temperate and they form a homoimmune series. We can readily infect our wild type strain, 10-fold serial dilutions, right down to individual plaques. These are the cluster N phages here, and we have a control phage Giles. So if we infect our wild type strain, we can recover cells from the infected area and show that they are lysogens. So this is an example of an MC² 155 butters lysogen. We know it's a lysogen because it confers immunity to infection by butters. It releases phage particles into the supernatant, and by PCR it has an integrated genome into the host chromosome. And we can form lysogens there for all of these cluster N phages, and we can show that the N lysogens are all immune to superinfection by the N phages as shown here. And in fact, we know that this is repressor mediated because if we clone the repressor for one of these phages, make a recombinant strain expressing that, then we reproduce the immune phenotype for all of the cluster N phages, but not a control phage, which in this case is Chi8. We've looked at gene expression in both lytic and lysogenic growth. When we look at infection by Michel-Mabel, isolating RNA 30 minutes or 150 minutes post-infection and look at the lytic gene expression patterns, we see in this strand-specific RNA-seq that the early genes are expressed early on, these rightwards facing genes here, and then at the later times as a transition to late lytic growth, these genes at the right end that go then through the ends back into the viral late lytic genes for virion structure and assembly. So that's the overall pattern. It's similar for Michel as it is for Charlie. If we look in lysogeny, so we simply grow lysogens of, for example, Michel-Mabel, isolate the RNA and map the RNA-seq reads back to the phage genome, you can see that the early and late lytic genes are all tightly switched off. But now that we see the repressor is being expressed as we would anticipate, but in addition there is a region of gene expression in this central portion, essentially between the lysis genes and the immunity cassette. And this is true for the Xerces and Pancino lysogens, all shown in this particular figure. If we focus in on the central part of each of these genomes, Michel-Mabel, Charlie, Fran, etc., you can see that all of these, even though there's diversity of the genes in this central portion, that they all exhibit these patterns of gene expression. Most of the genes in this region, we don't know what they do, but we know that for Pancino, for example, this strongly expressed gene here is predicted to be a single subunit restriction system that has sequence similarity to type 1 subunits. And because this restriction system we would predict is perhaps involved in conferring defense against viral attack, we wanted to test the hypothesis that this was the general property of what these different lysogens were doing. So what we did was we started out with these 10 lysogens shown by the columns here from Xerces through to Butters, and then we took 80 phages that span the spectrum of diversity of the mycobacteriophage collection, and we measured efficiency of plating of these attacking phages onto each of these lysogens, with the prophages providing the defense. And the efficiencies of plating are represented as a heat map, where the green represents no change in the efficiency of plating, and the red representing a reduction in the efficiency of plating by 10 to the 4 or greater reduction. So there's about 800 combinations of phage infections that we've tested here. About 10% of those show a reduced plating by 10 to the 4 or more. And although some of the lysogens, like pipsqueaks, defend against few of the attacking phages, other lysogens, such as Pancino, defends against a lot, perhaps over a third of all of those that we've tested. So this is a remarkable pattern of defense, suggesting that these various lysogens encode distinct types of defense systems that protect against phage infection. The patterns are informative and give some clues as to the genes that might be involved, at least in some of the instances. For example, the first 3 columns here are the phages Xerces, pipsqueaks, and Michel-Mabel. And I think you can see that Michel-Mabel and Xerces, the first and third columns, have closely related patterns, but that pipsqueaks is different. If we look at the genomes, we can see Michel-Mabel, pipsqueaks, and Xerces are extremely similar and closely related in this central portion of these genes, the ones that are expressed in lysogeny. The difference being that pipsqueaks has a 25 base deletion in the middle of this reading frame that is otherwise intact in Michel-Mabel and Xerces, and therefore we make the strong prediction that this gene, which is gene 29 in Michel-Mabal and its related version Xerces, is responsible for the defense that's being conferred as seen in this heat map. Similarly, we can see that Xeno, Skinny Pete, and Charlie have a similar pattern of defense, and they confer defense against a single attacking phage, which is the unrelated phage Chi9C, and again you can see Xeno, Skinny Pete, and Charlie are very similar within this region. So in total, we've identified at least 4 distinct defense systems in these phages, and these are the data that supports those. The first one is the restriction system encoded by Pancino. The second is this defense by the Charlie prophage against Chi9C, and we can see that this is mediated by gene 32 in Charlie, because if we knock it out, then we get no defense, and if we knock out other genes, you maintain the defense. And then if we clone gene 32, on a plasmid, it confers defense against infection. This gene 32 encodes a protein that's predicted to be a membrane protein, and so this single membrane protein could be a candidate for an exclusion type system that perhaps prevents DNA injection for proceeding with Chi9C, but with quite strong and quite striking specificity for this single unrelated phage. The other 2 systems are one that's encoded by Michel-Mabal, as I mentioned in the previous slide, and then the other is by a prophage called Fran, which protects against several phages, including Tweedy. So we have these 4 different systems where we can identify the genes that are involved. We're interested to try to find out what happens to the cells during the defense system. Are they committing suicide in an abortive infection type of system, or do they survive the infection? And we've done this assay by simply taking different strains, for example, a wild type strain or a lysogenic strain or various recombinant strains, and we take these, we grow them up in liquid cultures, and then simply plate out and look for bacterial growth on solid media that have been seeded with various different types of phages. For example, Tweedy, which is a temperate phage, or with a clear plaque mutant where we've deleted the repressor and is no longer able to form lysogens. So for our control strain MC2 155, we get high levels of survival, and these are lysogenic strains that survive the infection of the wild type Tweedy, but we get no recovery if we make this into a lytic phage that can no longer form lysogens. And now for our Fran lysogen, which defends against Tweedy, we can see that we still get high levels of lysogeny, but even if we knock out the repressor of this attacking phage Tweedy, we still get good levels of survival. So this is all consistent with the continued ability of the DNA of the attacking phage to be injected, and that the defense system acts in a post-DNA injection manner. So we were particularly intrigued by the Fran system. There appears to be 2 genes involved, 29 and 30. Gene 29 encodes a protein which has evident similarity to the PPGPP synthetase domain of Relay SPO-T-like proteins. Just as synthetase domain, it lacks the hydrolase domain or other related aspects that some of those proteins have. There is no sequence relationship between Michel-Mabal 29 and Fran 29, but they may have related functions given a complex set of relationships they have shared with a third phage called Squirty 29 that I won't explore in further detail here. For Fran 29, this PPG-PP synthetase, this gene cannot be transformed into Mycobacterium smegmatis alone. We have had no success in doing so, but we can do so easily if gene 30 is included. And just the 2 genes alone is sufficient to confer the defense phenotype. So a model for this is that the prophage, in this case we're talking about Fran, would express gene 29, shown by the gold, and 30, shown by this purple color, and that PPG-PP synthetase is kept in an inactive form by its partner protein, essentially acting as an inhibitor. And that what happens is that during a phage infection of phage like Tweedy, lytic growth happens, there is some type of activation which results in dissociation of these two, removal of the inhibitor, activation of the synthetase activity, synthesis of PPG-PP that then acts as an alarmone to cease cellular growth and phage development. We imagine that Michel-Mabel 29 and 30 acts in a similar way, although it may have some kind of distinct type of catalytic activity. Now, we've been interested in trying to find out how what the specificity is and how the phages trigger the defense system. And in order to try to address that question, we isolated defense escape mutants, or DEMs, in this case of phage Tweety, the attacking phage that is normally defended by Fran. So we could isolate independent mutants that can overcome Fran defense. Here they are, the DEM so-called 200 series, and these now plate with an efficiency plating of 1 on the Fran lysogen. When we map those, all of those have variations in an unusual Tweety gene called 54, and 54 is unusual in that it has a unique segment of its N and C termini, but these flank a tetrapeptide repeat, of which there's more than 40 copies, and each of the mutants, each of the DEM mutants, has a different number of repeats or variation in the actual repeat sequence itself. However, it's clear that these are defense escape mutants because if we simply isolate gene 54 that contains that repeat sequence, it remains fully subject to defense. And so these are therefore likely to be gain-of-function mutants which have acquired the ability to act counter to this Fran system. And so there's a specificity here to this counter defense system, which appears now to be able to tune itself so it can inactivate the or prevent the activation of the gene 29 and 30 pair of genes. And we've shown subsequently that we can tune this to act against other defense systems. So if this is simply a counter-defense system in gene 54, we can take a mutant where we've deleted it, this mutant here, and now isolate another series of mutants called the DEM 700 series of mutants, which have now overcome the Fran defense phenotype. And it turns out that these mutants now also overcome the Michel-Mabel defense system too. These map in several places, but most of them map to gene 57, which encodes a putative YB transcriptional regulator. However, all of these have nonsense mutations, and we don't know whether 57 itself is directly involved or whether it's other of the early genes that are expressed which are involved in this activation or this presumed activation of the PPG-PP synthetase. So we've described this series of at least 4 distinct defense systems, including the unusual use of PPG-PP synthetase as a phage defense system, and the Tweedy encoded counter defense system, which can tune itself to prevent those infections. We think that other mycobacteriophages and other bacteriophages, temperate phages in general, encode similar or novel types of defense systems, and therefore there is a rich resource to be tested and analyzed, with many new defense systems to be discovered. So I'd like to say a special thank you to all the folks in my lab who have contributed to this work and are authors on the paper, but also to the students and the faculty in the C-Phages community, and this is showing some of our colleagues at the annual symposium of the C-Phages program in 2016. If you have any questions, I would be delighted to answer them. Please send them to gfh@pitt.edu. Thank you, and I hope you enjoy the paper.